from our from amino acids, and then catabolism of the acids, and by synthesis of the non-essential amino acid, because the this one you need to take from the diet. Uh, the essential you need to take from diet, non-essential you can produced by our body, and then transport ammonia, then different process of urea cycle, then ammonia intoxication and disorders of the urea, and last but not least is the inborn errors of the amino acid metabolism. So a bit different on this part is that there are many inborn errors related to the amino acid metabolism, by means that when there's a defect of these amino acids, it can cause more. All of these are um, genetic disease, which is why we call it as inborn errors of amino acid metabolism. So when the babies are born, you can straight away detect there's all these symptoms, so you can detect the disease. Okay. So first we go to the digestion. So this one also is I think so I need to go through one by one because all these are inside my slides. And again, like uh, carbohydrate and lipid. So the digestion, where like carbohydrate and lipid, it starts at mouth, but this one protein starts in the stomach, so by the gastric uh, acid juice. So first of all is the pH. Okay, the acidic pH is the one that will activate the action of the pepsin. Okay, and then it will continue in the intestinal. So it will be released by the pancreatic enzymes and the intestinal enzymes. So these are the enzymes. Okay, you have the endopeptidase, exopeptidase, begins in the stomach. So the pH is the one that will activate the enzymes. Then the protein hydrolysis. So all these will hydrolyze the if you remember structure, okay, basically like enteropeptidase, it will cleave off the N terminal end of the trypsinogen and produce trypsin. Sorry. And then uh the trypsin in turn activates the trypsinogen. Oh, so this one is on the activation of the enzyme. Like I mentioned, all the enzymes are all in the inactive form. So HCl is the one that activates the enzyme and produce all these enteropeptidase so that it can activate the trypsinogen to become the trypsin. So trypsin in turn activates the trypsinogen and other zymogen. Whereas in the mm, intestine, the pancreatic sodium bicarbonate. Okay, this sodium bicarbonate will neutralize the acid contents from the stomach. So this is how it's being activate all the enzymes. So these are the enzymes that are in the stomach pancreas. Okay, so this is how it's being how the our protein are being hydrolyzed into smaller molecules, into small amino acids. Like for this one, the trypsin, it will cleave the protein, it will produce the arginine and lysine, and then chymotrypsin, it will produce this phenylalanine, methionine, so all these, so different enzymes, it will cleave at different sites to produce these specific amino acids to the enzymes. Okay, like carboxylase, it will cleave at the carboxylate site. Okay, so this carboxyl, if you still remember, carboxyl group, amino group. Okay, so that's the action of an enzyme, how it is being absorbed. Okay, it is absorbed same like the carbohydrate and lipid also. Lipid has transport, but amino acid no need for transport. So base transport. Uh, it can be through the facilitated diffusion, or it can be due to secondary active transport driven by the sodium gradient. Okay, so this, what are the questions? Okay, describe absorption. So absorption basically is just simple. Is absorbed through the glucose cells through this facilitated or secondary transport, and they have this amino acid carrier transport system. So this is a bit different with the some amino acids, they have its own carrier transport system. So if there's a defect to this transport, it can cause this heart nut disease. So like you have here large, like the small neutral amino acids, uh, it transport all these alanine, serine, threonine, and then the large neutral aromatic amino acids through this transport system, they transport all these. Okay, so these all amino acids are mainly the aromatic amino acids and the large amino acids. So if there's a defect on this transport system, it can cause this heart valve disease. So if you want to know more about the disease, you can check. But I'm only relating the if there's a defect to this transport system. So what kind of uh disease? So this is disease. What kind of disease it will cause? Okay. Okay. So that one is on the disease. There's a defect. Okay, briefly describe the following. So this one also is the special in the protein, because protein in our body, okay, our body always maintain a certain amount of nitrogen, okay, because mainly the amino acids are all in the form of the nitrogen, okay. So our body will maintain a constant amount. So if there's an excess of this nitrogen, it will be excreted through the urea, okay. So which is why in the protein is always related to the urea cycle. So what it means by the nitrogen balance is that the difference between total nitrogen intake and nitrogen loss is the same. So our body maintains this nitrogen balance. So for healthy adults is zero. Positive, meaning that there's a uh, excess or increase during the childhood growth. So when there's a building or a growth occurring, so it will have a nitrogen balance, so that all these amino acids can be used for the protein formation, enzymes, hormones, and all. So during it's positive during childhood growth, pregnant, muscle building, and also during healing. So during your healing process, it's also a positive nitrogen balance. It's negative when there's stress response, meaning when you are sick, when you are recovering from a um, not only from surgery, from a recall that from a disease that is a bit hard to heal, like even in COVID also says that person that already have COVID and is healing also have this nitrogen balance, because the recovery speed is reduced, and because COVID also has a high impact on the organs, okay, because it disrupt the other organ on the, okay. And other than that is when you have the malnutrition. So in kids that have malnutrition like kwashiorkor and all that, so they will also have this negative nitrogen balance. So it's the same. Okay. So our body maintains this nitrogen balance using this amino acid pool. So the human body uh, remain the amino acid pool at the 90 to 100 gram of amino acid. So this pool is supplied by the degradation of body protein. So meaning that when your protein degraded. So it can be in a form of like enzymes. So uh, some enzymes and some hormones also are degraded, and this will then supply to the amino acid pool. Then your diet, the protein that you diet, then the synthesis of the non-essential amino acids will contribute to the amino acid. When it's depleted by the synthesis of protein or drug, so synthesis by back
Doctor, I'm sorry to interrupt, but I think your mic is cutting off.
Oh, really?
Mm-hmm.
Okay.
It's better now.
Better.
It's better
Yeah,
now.
it's better now.
Okay.
Thank
Thank
you,
you.
doctor.
So this one, what was I saying just now? Oh yeah, the amino acid pool is depleted by all these, like synthesis of body protein and then all these conversion to glucose. When there's a gluconeogenesis, also this amino acid pool will be depleted. Okay, so this is in general how our body replenish this amino acid pool. Okay, and there's also this protein turnover. The total amounts of protein in the body remain constant. Okay, sufficient enough to replace all the protein that are degraded. So this one is called the protein turnover. So how they are being degraded is through this. Okay. There's a PAS, this called sequence. Okay. Recognize this proline, glutamate, serine, and threonine. So we call it as PAS sequence. It adds a signal peptide for protein degradation. So any protein that is bound to be degraded will be how call will be signaled by this will be bind by this PAS sequence, so that we know that the body knows that this protein is found to be degraded, so this ubiquitin protein system will bind to it. So it's a kind of protein. So this one. So this is the ubiquitin protein mechanism. So protein selected for the degradation is tagged with this molecule ubiquitin. Then the ubiquitin protein are recognized by the proteasome, and it will be hydrolyzed um, by this proteasome. So it will cleave all the protein into smaller amino acids. Okay, so this will happen. So ubiquitin proteasome mechanism, this ubiquitin will bound to the protein, and then this potassium will sort of lyse or degrade it into smaller amino acids. Okay, so in the end you produce the body will produce these smaller fragments amino acids. So these are the one that will contribute towards the amino acid pool. Okay. Okay, so I think that's all. Those are the things. Okay. So when there's a excess in the amino acid pool, then only our body will excrete the excess nitrogen. So the excretion of this nitrogen will be through the urea cycle. So before we go towards the urea cycle, so we go towards the amino acid metabolism first. So in summary, we've already gone through the carbohydrates, okay, fatty acid. So now we go towards the protein. So we'll see how the proteins are being metabolized. So the main thing or the main reaction that you need to remember is there are two reactions, main reactions in protein metabolism, mainly the transamination and oxidative deamination. Okay. So this is the reaction. Okay, transamination. Identify reaction. Okay, this is transamination. What happens to ammonia in this reaction? Hmm. In transamination, what happened to the ammonia? So this is the ammonia group, NH3. What happened to ammonia in this reaction? What happened to ammonia in this reaction? Transamination process.
It is removed.
Remove.
Yeah, transfer into glutamate.
Yeah, basically it's a transfer of the ammonia group. Okay. As the name implies, the transamination is a transfer of this amino group. So amino transfer is increased by while aspartate. Okay, these two enzymes are example of which reaction, right? Okay, so this one, alanine aminotransferase and aspartate aminotransferase also is the main enzyme in the transamination process. Okay. Transamination. Okay, and liver is the main site of the transamination. So all this process occurs in the mainly in the liver. Okay, aspartate alpha ketoglutarate. So this one is a aspartate aminotransferase. So uh, another thing is that it has this coenzyme. Okay, predoxal phosphate. It will receive the amino group predoxamine. It will converts to predoxamine phosphate. So this is how it's being transferred, the amino group. So without this predoxal phosphate, the reaction transamination cannot occur. So it's important coenzyme or cofactor for this transamination. AST, ALT, AST is aspartate plus alpha ketoglutarate. So we produce the oxalate, oxaloacetate and glutamate. Whereas in the alanine aminotransferase, so alanine plus alpha ketoglutarate will produce pyruvate and the glutamate. So these are the main reactions of this transamination. So if you still remember in the Krebs cycle, okay, Krebs cycle, that you have this alpha ketoglutarate especially, so it means that all like the aspartate can like this one aspartate can be converted to the oxaloacetate. That's the Krebs cycle here, right? Okay, so this one, okay, like aspartate, where's aspartate is now? Okay, aspartate can be converted to the oxaloacetate. So aspartate can enter the oxaloacetate, can enter the citric acid cycle through the oxaloacetate. Like alanine is converted to the pyruvate. Yeah, here, this one. So all these, so it means that all these amino acids are the ones that can be converted to in the gluconeogenesis. So all these. Whereas you have this glucogenic and the ketogenic acid. So glucogenic amino acid can be converted to glucose, whereas ketogenic amino acid cannot be converted to glucose, but it can be converted to ketone bodies. Okay, where am I just now? Oh, okay, transamination. Okay, so these are the main. So next one is on the oxidative deamination. Okay, produce of phosphate. Okay. The reaction, transamination reaction is reversible. Okay, so it can go both way. So it's reversible transamination. Same like same like this oxidative deamination is also reversible. This one oxidative deamination is mainly conversion of glutamate to the alpha ketoglutarate. So this one is like transamination is transfer of amino acid So this one is it release or it accept the NH3 ammonia. So the main enzyme is the glutamate dehydrogenase. Okay, and it requires this NAD plus and NADP plus as a coenzyme. Just now it requires the predorsal phosphate. This one NAD plus or the NADP plus. Okay, and then the reactions also depends on the concentrations of the glutamate. Usually this reaction occurs primarily in the liver and the kidney, and it also occurs in the muscle, in the other tissues. Okay, when you want to transport the ammonia to be excreted out of our body, okay, through the glucose alanine cycle. So we go through that later. So it's a kind of a deamination, as the name implies, oxidative deamination, but it can also receive the amino group. Okay, any question on this too? So these are the two main reactions that you need to know in the amino acid metabolism. These are the main reaction that occurs: transamination and the oxidative deamination. Oh, state the allosteric regulators for glutamate dehydrogenase. So this enzyme also is being highly regulated. Okay, so you have here GTP, guanosine triphosphate, is the allosteric inhibitor for the glutamate dehydrogenase, whereas the adenosine diphosphate ADP is the activator. So these two are the main inhibitor and the activator for the glutamate dehydrogenase. What else the question? Okay, that's all the question. Which group is presenting and which activity?
M14 will present activity 5.
Okay, activity 5. And the other one?
M17 will present activity 7.
Okay, activity 5. Okay. So any question on the transformation, oxidative deamination? So that's basically the enzyme reactions involved. And then, like I mentioned just now, you have this glucogenic and the ketogenic amino acid. Well, basically, these two, there are some that are glucogenic both, glucogenic and the ketogenic amino acid, but only leucine and threonine, I guess. Leucine and lysine. Leucine and lysine are strictly ketogenic. This one. Leucine and lysine. Leucine and lysine. So these two can be converted to acetyl-CoA and also acetyl-CoA. This one is strictly ketogenic, whereas the rest can be converted to glucose. So it's glucogenic amino acid. Okay, so these are the amino acids that can be converted to glucose by this gluconeogenesis. Whereas ketogenic amino acid, leucine and lysine can be catabolized for energy in Krebs cycle or converted to ketone bodies or fatty acids. Okay, because these can be converted to acetyl-CoA and the acetyl-CoA. So acetyl-CoA can be converted to ketone bodies or the fatty acids. Okay, any question on this? So these are the, okay, again, same. Glucogenic amino acid. Okay, this is showing one by one the pyruvate, how the amino acids are being converted. So you have this pyruvate entry point, and then the oxaloacetate, succinyl A entry point. What else? So that's the main thing. Okay, state the importance of folic acid in catabolism of methionine and its associated disease. So what's the importance of folic acid in this? In amino acid metabolism? In which one? In pyruvate or in oxalic acetate? Which one is important? At which reaction that this folic acid is important?
Succinyl CoA.
Okay, the succinyl CoA entry point. I hope you can see this. Well, you have your own slides, right? So this folic acid here, methylcobalamin, and also this cysteine, say B6, B12, and B6. Okay, so for this reaction, it requires this vitamin B12 and B6 or the folic acid. Okay, so without these cofactors, this reaction cannot occur. Okay, what's so important about this reaction is that methionine, okay, what the amino acid, okay, it will be converted to this SAM as adenosine methionine. So then it will be converted to SAH as adenosine homocysteine, and then to the homocysteine. But the thing is, without the folic acid, vitamin B6 and B12, it cannot be further converted because this homocysteine is actually toxic to our body. So when there's presence of this homocysteine, our body will try to eradicate it, not eradicate, but convert it. It's either back to the L-methionine. So by using this reaction, so homocysteine plus the tetrahydrofolate, or tetrahydrofolate also is one of the folic acid. Okay, homocysteine plus the tetrahydrofolate with this cofactor methylcobalamin, it will produce back L-methionine and the tetrahydrofolate. And on the other hand, homocysteine also can be converted to the cystathionine, okay, by this cystathionine beta synthase to produce the cysteine. Okay, so this is how cysteine are being produced. So without this, accumulation of homocysteine is you can say that it's quite detrimental, which is why when you have this, especially in a pregnant woman, okay, elevated of homocysteine or decrease of folic acid, these can cause the neurotube defects, which is why for a pregnant lady, we advise them to take folic acid as earlier as possible, okay, to decrease the incidence of this neurotube defect. So it is an improper closure of the, as in the spina bifida in the fetus, mainly because of this accumulation of the homocysteine. Okay. When there's an increase in this homocysteine, it can cause oxidative damage, inflammation, endothelial dysfunction. Okay, so which is why homocysteine level is usually, oh, the body will try to eradicate as soon as possible. Okay, any question on this? So which is why folic acid, vitamin, all these cofactors are important in order to convert the homocysteine so that it's not toxic to the cells. And another thing is that this one, as an adenosine methionine, so it's also one of the major methyl group donor in the biological reaction. So it's important in the phospholipid synthesis, even the nucleotide synthesis and the synthesis of the amino acid. It's also important in the reaction like post-translational protein and the DNA methylation. So that's the disease related to the especially the succinate A entry point. Then other than that, okay, so this one you can just read. When there's a this one is also important aromatic amino acids because this one when there's a defect of this phenylalanine hydroxylase, okay, as you know that phenylalanine relate to the which disease? If there's a defect of phenylalanine, it can cause what disease? Which inborn error is related to phenylalanine? Which one? Which disease is related to this phenylalanine?
Phenylketonuria.
Okay, so phenylketonuria. So without this phenylalanine hydroxylase, phenylalanine cannot be converted. Okay, so that will cause the phenylketonuria. And then these are also other disease related to it, like tyrosinemia, alkaptonuria. Okay, PKU and the alkaptonuria I mentioned in the inborn errors of the metabolism. Okay, so which is why these enzymes are all important. So if there's a defect of this enzyme, it will cause disease. Okay, what else? So all these the like oxaloacetate, entry point, okay, have the asparagine and then the aspartate I mentioned just now. Aspartate can be converted to oxaloacetate. So by this transamination, pyruvate. So these are the alanine can also be converted to pyruvate through the alanine aminotransferase. Okay, so this the other reaction, and then glycine serine also can enter the pyruvate. Okay, so I hope you can remember all this. So this one is on the okay, just go straight here. Amino acids biosynthesis. With some amino acids have to be synthesized by our body. Okay, like you have here, like tyrosine, okay, is synthesized from phenylalanine, then glutamate, glutamine synthesized from glutamate, and then this proline and arginine also synthesized from the glutamate semialdehyde. So all these, okay, origin of the non-essential amino acids. So meaning that all these are non-essential amino acids, meaning that all these amino acids are being produced by our body. Okay. So, okay. Yeah, that's just straightforward. Any question on this? So meaning that other than these amino acids are being utilized, okay, by meaning utilized is that this is how it is being utilized. This is how it's being utilized, okay. So meaning that it enters the Krebs cycle, it's either being used for gluconeogenesis or it's being utilized as in it's being converted to other substance, like in this case, succinate A being converted to cysteine, okay, and the production of the SAM as adenosine, and the other one is the product biosynthesis of the amino acid. So in amino acid metabolism, there are three important things. Okay, so it's either to produce back the glucose through the gluconeogenesis or the ketone bodies. Then means that to provide energy. The other one important is to produce other intermediates that are important for reaction. and the other one is for the amino acid biosynthesis. So these are the non-essential amino acids that are being produced by the amino acid metabolism. And then the last but not least is for the uh, excretion of the ammonia. So before excretion of the ammonia, so that's like I mentioned just now, our body maintains the amino acid pool. So all these is either being utilized, enters the amino acid pool, is being utilized. Unlike the glucose, carbohydrate, fatty acid, all of that can be stored in the form of fats. But for ammonia or for amino acids, it cannot be stored in our body. So it's just temporarily be there in the amino acid pool. Uh, if there's an excess, it will be excreted to recycle. Because ammonia is considered is also toxic to our body. There's an increase of the ammonia, hypoammonemia, hyperammonemia. So that can cause a lot of okay, we mentioned later. Okay. Any question on that? Hope you're clear on all those, especially on the importance of the folic acid and relating to the disease. Okay. Before I go further, so this is how our body transport the um, ammonia. Okay. Uh, Ammonia mainly in the most tissues and also the muscle. Uh, Basically, the muscles is the one that has a lot of amino acids. Okay, is the one that produce the most ammonia, mainly because of the protein in the muscle. Okay, some some of these protein are degraded when don't use the muscle or when there's fasting in the during the fasting state. These uh, protein will be catabolized to produce energy. So all these ammonia will be transported this glucose alanine cycle. All this is called the glucose alanine cycle. So all process from muscle, from other tissue, most tissues like other organs like spleen and all. This is how it's being transported. Mm. So mainly through this one, you can see this. So from most tissue, so what is the reaction? This one, transamination or oxidative deamination? What kind of reaction is that? There's glutamate and then glutamate synthase. Transamination or oxidative deamination? This one is still considered as the oxidative deamination, although the glutamate synthase, because this one received the um, ammonia. Okay. And then it will be converted to the glutamine. Hmm. Okay, glutamine, all the glutamate will be converted to the all the glutamate will be converted to the glutamine. This is one. Where is it mentioned? Oh yeah, here. So all glutamate will be converted to the glutamine because it's a non-toxic transport from ammonia and most important transport to form of amino nitrogen in blood. Okay, so NH3. Okay, it cannot be transport just like that in the mean that it cannot be released into the blood vessel that I mentioned that ammonia itself is toxic. So it is converted to the glutamine in order for it to be transported to the liver. And in the liver, it will be converted back to the ammonia. Okay, so this reaction occurs in the most tissue and same in the muscle. Okay, this one is what? Transformation or oxidative deamination. Oxidative deamination, which see the glutamate oxidative deamination, and then the glutamate convert. Alanine is a transformation. So this one occurs in the muscle. So again, uh, glutamine from other tissue, whereas alanine the muscle. So that's the difference between the muscle and the other tissues. So other tissues are very whereas uh,
Doctor, can you please repeat the uh, can you please repeat because your mic just tripping just now.
tricky. Okay, which one? The muscle or the most tissue?
Uh, the one you try to summarize both.
Uh, Summarize. Okay. Okay, I just repeat back. So in the muscle, all these amino acids break down to the ammonia, NH3. Okay. Oxidative deamination, we convert it to the glutamate, and then glutamate to transamination, it will convert to the alanine. Okay, so this alanine in the muscle, it is all the ammonia is transported by this alanine. Whereas in the most tissues, the glutamate are all converted to the glutamine. So this one oxidative deamination and then glutamine synthase, you convert it to the glutamine. So in most tissue, it's transported through the glutamine, whereas in the muscle, transported through the alanine. Okay, is that clear? Okay, so this glutamine will go to will enter the blood vessel to the liver. So in the liver, it will be converted back to the NH3. So this NH3 will goes to urea. Okay, any question on that? So this one also straightforward. Glutamine converted back to the ammonia. So this one is straightforward. So this reaction also involve the oxidative deamination. Okay. Whereas in this one, in muscle, it is called the glucose alanine cycle. The glucose alanine cycle here is that alanine will go to the liver. So this one is the same thing, liver. It's just that I cannot put it together because it's long. So alanine will go to the liver, so we convert it back to the pyruvate. So by this transamination, so it will produce the glutamate oxidative deamination, it will produce the ammonia. So it cycles back. So I mean pyruvate, it will convert to glucose. Glucose goes to the muscle. Okay. So glucose will be converted to pyruvate so that this ammonia can be converted to alanine. So this is the glucose alanine cycle. So this only occurs between muscle and the liver. So mainly this is to transport the ammonia. So NH3 later we use for the urea cycle. Any question? So I hope you're clear on that. That's why I said oxidative deamination and the transamination is the two reactions that is important. So mainly, yeah, uh, in the transfer of ammonia, mainly these two reactions are important. Other than this conversion to glutamate to glutamine by this glutamine synthase and the glutaminase. Any question? So I hope you're clear on that, on the transport of ammonia. So I've discussed this one. So if there's no question, um, which group represent the activity 5? Can present now.
Assalamualaikum warahmatullahi wabarakatuh, and a very good morning to Dr. Fai and my fellow classmates. Today, my group M14 will be presenting amino acid metabolism activity 5. So, next slide please. Okay, here the statement says that urea is a major disposal form of amino group derived from amino acids and accounts for about 90% of the nitrogen containing components of urine. One nitrogen of urea molecule is supplied by free ammonia and the other nitrogen by aspartate. Okay, so we head on to the first question. First question is draw the structure of ammonia and urea. So ammonia has the chemical formula of NH3. So the structure of ammonia will be the picture on the left, and urea, which has the chemical formula of CH4N2O, has the chemical structure has the structure on the picture on your right. So the next question, I will pass it to my teammates. Thank you.
Assalamualaikum and good morning. So for the second question, it states that it wants us to state the sources of ammonia in the body. So there are 5 sources. First is from the amino acids. Many tissues, especially the liver, will form ammonia from amino acids through the process known as the deamination, which is through the linking of aminotransferase and glutamate dehydrogenase reaction will actually produce ammonia. Second is from the glutamine in kidney will produce ammonia and glutamic acid by glutaminase enzyme. And then the third will be from the bacterial action in the intestine. So ammonia will be produced by the action of intestinal bacteria on the non-absorbed dietary amino acids. Fourth would be from the amines. Ammonia is released from monoamines by the action of monoamine oxidase enzyme. And then lastly will be from the purines and pyrimidines, where ammonia will be released during purine and pyrimidine catabolism. That is all. I will pass to the next presenter. Thank you.
Okay, so for the third question, draw the figure of the urea cycle. So it's drawn and let me explain. The urea cycle predominantly occurs in the hepatocytes. Urea cycle is a process which basically converts the toxic ammonia into a less toxic form, the urea, to be excreted out of the body. So in the mitochondria, the excess ammonium combines with carbon dioxide that actually originates from the bicarbonate, and therefore using two ATP molecules to form the carbamoyl phosphate. At the same time, take a look at the fifth step, where ornithine is produced. So now the ornithine moves to the mitochondria, combines with carbamoyl phosphate to form the citrulline. Then the citrulline moves to the cytoplasm, combines with the aspartate to form the argininosuccinate. The argininosuccinate is then broken down into arginine and fumarate. In the final step, the arginine is hydrolyzed into ornithine and the cycle repeats. Yeah, that's about it. Thank you.
Okay, moving on to the next question. Okay, hello and good morning. So, question 4 is explain the reactions that occur in urea cycle. So now we understand what is the importance of this urea cycle. I will be explaining the main steps that occur in the urea cycle. Okay, so there are 5 steps that we need to understand. But before I begin explaining them one by one, you need to understand that urea cycle occurs in the liver cells. So the first 2 steps will take place in the mitochondria of the liver cells, and the remaining 3 steps occur in the cytoplasm of the liver cells. Okay, can we refer to the diagram shared by Janessa just now? Okay, Harmit, can you go to the previous slide, please? Harmit, can you go to the previous slide, please? Okay, thank you. Okay, so if you guys can see, in this diagram, the first step occurs in the mitochondrial matrix. So the combination of carbon dioxide and ammonium ions produces carbonyl phosphate. So this reaction, it requires 2 ATP molecules and an enzyme carbonyl phosphate, synthase 1. Okay, so after the formation of carbonyl phosphate, it will combine with ornithine. Okay, so if you guys can see, ornithine is actually the product of the last step in the urea cycle. So this ornithine is used to combine with carbonyl phosphate to produce citrulline. Okay, so this reaction is catalyzed by the enzyme cornithine transcarbamylase. Okay, so in the third step, citrulline, which was produced in step 2, is transported into the cytoplasm. Okay, so in step 3 is already occurring in the cytoplasm, not in the mitochondrial matrix anymore. Okay, so citrulline will combine with aspartate, which is an amino acid, and producing arginosuccinate. Okay, so this reaction is catalyzed by the enzyme arginosuccinate synthase, and this reaction requires 1 ATP molecule. Okay, so in the fourth step, arginosuccinase, which is an enzyme, acts upon arginosuccinate. Arginosuccinate was produced by step 3. So arginosuccinase acts upon arginosuccinate, producing 2 molecules, which is arginine and the fumarate. Okay, if you guys can see, the arrow for fumarate is going outwards. Okay, so this is because fumarate won't be used for the next steps of urea cycle. Fumarate will be hydrated to malate, which is used for other metabolic pathways, for example, gluconeogenesis. Okay, whereas the arginine that's produced will be used for the next step of this urea cycle. Okay, so the last step, which is the fifth step, the enzyme arginase acts on the arginine and lyses it to produce urea and ornithine. So remember ornithine from our second step. This ornithine will be used back for the next cycle, whereas the urea, which is soluble compound, it dissolves into the blood and it transports into the kidney to be filtered and excreted in the form of urine. Okay, so that is all for urea cycle. Thank you so much.
All right, so I'll be explaining question 5. Okay, so I just added a diagram so it'll be easier to understand. Okay, so the main enzyme that is involved in the regulation of urea is CPS1, also known as carbamoyl phosphate synthetase 1. It is regulated by N-acetyl glutamate, which is a positive modifier for this enzyme. N-acetyl glutamate is synthesized from acetyl-CoA and glutamate, and is stimulated by an acetylglutamate synthetase for which arginine is the activator. So basically, when there is more protein degradation, more ammonia is produced. That means there will be an increase in glutamate. This increase stimulates CPS1 to form more carbamoyl phosphate and activates the urea cycle. So that's it from me.
Other regulations of urea cycle?
Yeah, I just explained it.
Other than the carbamoyl phosphate synthetase.
And N-acetylglutamate, right?
Yes, okay, that's correct. Other than that?
I'm not sure.
The regulation is by the main enzyme, okay. All that you explained is correct. Is there other than that? Any other condition that can cause the urea cycle to be activated or inhibited?
I'm not sure.
Is it inhibited by acidosis?
Yes. How is it inhibited by acidosis? How is urea cycle inhibit? How acidosis inhibit urea cycle? In acidosis, what is SS? Hydrogen or bicarbonate?
Hydrogen.
Yeah, hydrogen is in excess. So in order to reduce or to balance the excess hydrogen, what will happen? What will happen? This is basic foundation knowledge. In order for you to reduce the acidosis, the excess hydrogen, you need. How did you balance the excess hydrogen? Yeah, by the
Carbonate.
bicarbonate. Okay, so which is why bicarbonate is also used in the urea cycle. So which is why urea cycle is inhibited because all the bicarbonate is being used to balance the hydrogen. Okay, so that's another condition that regulates the urea cycle. Okay, another question is that how is this is urea cycle can interconnect with the Krebs cycle? Urea cycle and Krebs cycle. Can or cannot?
It can. It can.
Yes, it can. So where? Where it is connected? Interconnected.
Fumarate. Fumarate.
Fumarate?
Will be used to form malate in the Krebs cycle.
Malate in the Krebs cycle. Okay, so all these, okay, you're correct. Fumarate, malate, and also the oxaloacetate. Oxaloacetate by which one? How is oxaloacetate is can be connected to the urea cycle? Your friend did mention just now.
Isn't it from malate to
Mm,
oxaloacetate?
malate to oxaloacetate. Okay, oxaloacetate. Uh, what kind of amino acid is interconnected between these two? Oxaloacetate and the
Transamination of from aspartate, right?
yes, aspartate. Okay. Because oxaloacetate can be converted to aspartate or aspartate to oxaloacetate. So this aspartate can enter the urea cycle through what? Or the aspartate can be produced through what reaction in the urea cycle? Yeah, arginosuccinate. Okay. The reaction to produce the arginosuccinate will produce the aspartate. So this aspartate is one of the your friend did mention just now that this aspartate can be further used for gluconeogenesis, right? So this aspartate can enter the Krebs cycle through the oxaloacetate and can be used for synthesis of amino acid or the gluconeogenesis. Okay. Any question on this group? Okay. Thank you. Good presentation. Any question? So I hope you understand on all the processes of the urea cycle and how it is connected to the Krebs cycle and the regulation. Okay. Okay. Moving on. So this. So what happens when there's a increase in the ammonia? Like I mentioned just now, when there's a increase, it is toxic to our body. Okay. These are types of hyperammonia. So all of these are here. What's the question? What will happen? Oh, okay. If there's a liver disease, what happened to the blood ammonia? Okay. So basically, when there's a liver disease, what will happen? Why, if there's a liver disease, ammonia can increase? What do you think will happen? Basically, because liver is one that the main organ that will the urea cycle is happening is in the liver. So if there's a liver disease, urea cycle cannot happen. So this urea will be spilled into the blood vessel. So this excess urea, we call it or urea pula dah, ammonia. So this excess ammonia, because ammonia cannot be converted to urea to be excreted. So all this ammonia will be spilled to the blood vessel. So it will cause hyperammonemia. So the same, hyper means increase, ammonia, anemia means that it's in the blood. Okay, hyperammonemia. So high toxicity can cause this tremors, slurring of speech, vomiting, cerebral edema, blurring of vision. So if it's a very high concentrations of ammonia, it can cause coma and death. Okay, it's the same also if you have kidney disease. Okay, but kidney mainly is urea, hyper, increase of urea in the blood vessels. So that one is a bit different. Okay, increase of urea. Increase of urea also can cause almost the same, but basically you can see the like what do you call it? Because urea is a waste product, so you will have like skin itchiness. Okay, because all this urea will deposit on skin, so it can cause itchiness and all. So it is a bit different, but still high. If there's a very high level of urea, it can cause all these mm, vomiting. Okay. So these are the types of the hyperammonemia. So you have two types here. Basically, we divide into two. It's either acquired or congenital, meaning that congenital is a genetic disorders. Acquired meaning it's due to secondary of other disease such as liver failure, um, chance operation, okay, elevated levels of circulating ammonia. So all these are due to the secondary is due to other disease, whereas congenital hyperammonemia is due to defect of the enzymes. So this is a genetic defect. So hyperammonemia type 1 is due to carbamyl phosphate synthetase 1. Hyperammonemia type 2 is due to ornithine transcarbamylase enzyme, but most of the time it's due to this carbamyl phosphate synthetase. Okay, what is the enzyme? Oh, state the normal value, state clinical symptoms already. Describe the mechanism of ammonia intoxication. Oh, the normal value and all is all here. Normal value is between the 5 to 50 micromole. Whereas if there's a hyperammonemia, it can increase up to 1000 micromole. Okay. What else? Oh, I've already describing this one. So congenital hyperammonemia, clinical symptoms and all is the same, this one. Blurring of vision and all. And if very high, it can cause coma and death. So these are the two types of the hyperammonemia treatment. Usually is by limiting the protein diet, because due to this defect of the enzyme, so urea cannot be produced. So we have to limit the protein in the patient's diet. So basically by administering the compounds that bind covalent to amino acid. Example, phenylbutyrate given already is converted to phenylacetate, because this phenylacetate is non-toxic and can be easily excreted in the urine, rather than it has to go through urea cycle and all, because this one can straight away be excreted in the urine. Okay, so this is the mechanism of the ammonia intoxication. Basically, this ammonia will deplete the alpha-glutaric acid from the brain. Why it depletes the alpha-glutaric acid from the brain? Basically, this one is on the oxidative deamination, right? All the glutamate will bind to the alpha ketoglutarate to produce the glutamate. Okay, so this will depletes the alpha ketoglutarate acid from brain. So this leads to the inhibition, because alpha ketoglutarate glutarate is also important for the Krebs cycle. So when it depletes, Krebs cycles in the brain cannot continue. So there will be reduced energy in the brain. So the brain is deprived from its main source of energy and leading to the neurological manifestation. So which is why you have all those symptoms just now, like the blurring of vision, vomiting and all. Okay. and the edema. Edema is it? Cerebral edema. So a slurring of speech and then a tremor. So all these because there's not enough energy production in the brain. So that's the mechanism. Any question on this? So I've answered all of this. Okay, so that's the main thing that you need to know on the hyperammonemia and what's the effect and how it is, what's the mechanism. So basically it just depletes the alpha ketoglutarate in the brain. So the brain cannot produce enough energy via the Krebs cycle and the ETC. Any question? If there's no question, we proceed to next group is activity 7, right? Okay.
Yes, doctor. M17.
All right. So you can present now.
So, good afternoon to Dr. Fai and my fellow batchmates. Today, M17 will be presenting activity 7. So, the case is as follows. A 19-year-old man with learning difficulties presents to the emergency department with sudden blurring of his vision in the left eye. He lives in a community house with other individuals with learning difficulties. He has a past history of deep vein thrombosis some 3 years ago, lens dislocation, severe myopia. On examination, he has a body mass index of 19 kg and a blood pressure of 135/78 mm. So you could also see that he is abnormally tall and has abnormally long leg length and also libido type appearance to his skin. So the first question asks us to state the diagnosis. So this patient is suffering from homocystinuria, and the other symptoms are mainly the clinical manifestation of that diagnosis itself, which is lens dislocation, hypertension, libido reticularis, and mild anemia. So on the top left, you can see lens dislocation. Mainly in homocystinuria, the lens dislocation is downward. And the middle picture shows libido reticularis, which is a reddish-blue discoloration of the skin linked to swollen blood vessels. And on the most right picture, you can see that this individual is suffering with the condition, tends to be tall in stature, abnormally long hand length, and osteoporosis. Now I'll pass to my teammate Vidya to explain further on this disease.
Good morning. As per mentioned above, the disease name is homocystinuria, which is a rare but potentially serious inherited condition, also known as autosomal recessive illness, whereby our body is unable to break down protein from the food we consume. And this causes a harmful buildup of substances in the blood plasma and in the urine. Next slide, please. So basically, there is some defects in the metabolism of cysteine, whereby a patient having this disease lack the enzyme cystathionin B synthase, which converts homocysteine to cystathionin. As a result, there will be high accumulation of homocysteine and methionine in the blood plasma and in the urine. However, there will be low level of cysteine because the production of cystathionin is inhibited. That's it for my part. Thank you.
Good morning. The treatment or the management for this patient is there is no cure for this disorder, but half people who are diagnosed with this condition show improvement from vitamin supplements may include therapy with pyridoxine, vitamin B6, a diet that restricts the intake of protein and methionine, betaine therapy and supplementation with folate, vitamin B9, or cobalamin, vitamin B12. Affected individuals may first undergo a pyridoxine response assessment. So that's all from our group. Thank you.
Okay, thank you. So any question for this group? Okay, no question. Okay, thank you, M17. Good presentation. So just oh wait. Okay. Before we go on further with activity 8, can you please sign in the attendance? Question, name the enzyme that is defect in the homocystinuria. Okay, so basically all these inborn errors, are, you call it as, it's a rare disease, okay? Okay. So all the inborn errors is a rare disease and is quite rare. Okay, all these are rare. But in Malaysia, so this one is based on the data that I have 2010, quite 10 years ago. So even if it's rare, but these are the usually that is we often see in the disease in the Malaysia. Well, in other countries probably is also different. So based on the genetic susceptibility and all. So especially in these inborn errors of amino acid, PKU, phenylketonuria, and maple syrup urine disease. These two are the main disease that we often see in the in Malaysia. Other than that, other like carbohydrate metabolism, galactosemia. Okay, still remember. Due to galactose pathway, there's a defect in the enzyme in the galactose pathway. Then fatty acid oxidation due to this, lysosomal storage, and then urea cycle that just now on transcarbamylase deficiency. Okay. So these are the common among the rare disorders in Malaysia. Okay, so this one, if you can see, is all the inborn errors that are related to the amino acid. But the one that I gave here is the one that is common among the rest. So phenylketonuria, MSU, captonuria, MSUD. Okay, like your friend present just now, homocystinuria. Then you also have the albinism. Okay, back to the question. Okay, early diagnosis of phenylketonuria is important because the disease is treatable by dietary means. So how do you state the importance of this early diagnosis of the PKU? So how do you detect early PKU? Is through this neonatal screening and the diagnosis of the PKU. So early diagnosis is important because the disease is actually treatable by the dietary means, meaning that you can't really treat the defect enzyme, but by giving strict diet to this fetus or baby, you can reduce the symptom. Okay. Basically, in this, there are two types. mainly is due to this phenylalanine hydroxylase. So when this phenylalanine hydroxylase is not present, so it cannot convert the phenylalanine to the tyrosine. So it's a genetic deficiency of the phenylalanine hydroxylase. Okay, when this defect, tyrosine cannot be produced, and then phenylalanine and phenylpyruvate is accumulated in blood. So this results in the mental retardation. So what's important about this tyrosine is that tyrosine is the precursor for synthesis of melanin. If you remember, melanin is important for skin, the coloration of your skin, melanins, and the epinephrine or epinephrine, okay, your neurotransmitters. Then catecholamines also. So which is why because of this no melanin this is being produced, so individuals with phenylketonuria have light skin and hair color, but it's not totally like albinism. Okay, is the most common among the rare inborn errors. So first one is due to this phenylalanine hydroxylase. Second one, it can be due to this deficiency of several enzymes that synthesize this BH4 or dihydroxypyridine reductase. Okay, so this is what happened. So deficiency in the dihydroxypyridine reductase or any of the enzymes of the BH4 leads to phenylalaninemia. Okay, and decrease the synthesis of the catecholamines and the serotonin. So when there's a defect on this BH4 production, so all these tyrosine, catecholamines, and serotonin also cannot be produced. Okay, so this second cause of the phenylketonuria, but this one is very rare. The most common one is due to the defect of the phenylalanine hydroxylase enzyme. State signs and symptoms. So you have all the signs and symptoms here, like the skin coloration, and then okay. Due to this elevated phenylalanine, because it cannot be converted to tyrosine, so you have accumulation of this phenylactate, phenylacetate, and phenylpyruvate, which are not normally produced. So this gives the urine musty or mousy odor. And then other than that is the mental retardation, again because of accumulation of all the phenylpyruvate just now. So this is quite toxic to the cell. So failure to walk or talk, seizures, hyperactivity, tremor, and rarely achieves the IQ greater than 50. So which is why early detection is important, so that we can give earlier strict dietary treatment, so that to avoid this kind of symptoms. Okay. Again, the hypopigmentation because of no tyrosine is being produced, so it has a fair hair and light skin colors. Okay, so treatment is by limiting the phenylalanine intake. So to levels are barely adequate to support the growth. So we give the tyrosine, so essential nutrient for individuals with phenylketonuria. So mainly we give the tyrosine diet and limit the phenylalanine intake. So actually, this condition is not that bad if you can have an early diagnosis and the treatment is given earlier. Okay, any question on this phenylketonuria? I think the rest of this you can read. Okay, like acatonuria is due to this deficiency of the homogeneous acid oxidase cause accumulation of this homogeneous acid. This also can be detect in the urine. Like just now, PKU, you have the mousy odor. This one, you have this dark pigment with homogeneous acid. oxidize to a dark pigment on standing. So this one. So if we leave it for 2 hours, the urine will turn entirely black. It's due to this homogeneous acid. So oxidation. So what happened is that this patient will have a large joint arthritis and then this black pigmentation in the cartilage and the collagenous tissue. So this one. So there's a black pigment in the cartilage of the tissue. Same, usually the treatment is basically to reduce or to limit to a certain diet, amino acid diet. So diets low in protein, especially in phenylalanine and also the tyrosine. So usually this patient with alkaptonuria, the difference with the alkaptonuria is that all other disease are detected as early as during the fetus or during the infant, but this alkaptonuria usually asymptomatic until about the age of 40. At the age of 40, then only you can detect the disease because it's not life-threatening. Only that when it's age, so it will cause the joints, so it will be harder for you to move and all. Okay. So that's alkaptonuria. So same with MSUD, as the name implies. How you the signs mainly is the order of maple syrup or burnt sugar in the patient. Okay, these amino acids. This is due to the partial or complete deficiency in the branch chain alpha keto acid dehydrogenase. So mainly this enzyme is important for leucine, isoleucine, and valine uh, to break down this leucine, isoleucine, and valine. So without this leucine, isoleucine, and valine will be accumulated. Again, accumulated will cause a toxic effect and interferes with the brain. So this is what happened. So valine, isoleucine, and leucine cannot be metabolized. Okay, accumulation of these will cause the all the symptoms. Okay, so these are the other symptoms. Have the feeding problem, vomiting, dehydration, and if it's untreated, it still can, like the PKU, it can lead to mental retardation and even death. And so the treatment, early diagnosis also is important. So treatment is giving a synthetic formula that contains limited amounts of these, all the branching amino acid. So leucine, isoleucine, and valine. So just to provide a sufficient to provide the branch amino acids necessary for normal growth and development without producing the toxic levels. Okay, homocystinuria your friend has mentioned, and then the albinism. Okay, is a complete no tyrosine. Is this one is due to there's no tyrosine production. Okay. Okay. Any question on this? All the inborn errors. So just a few of the inborn errors. If you want to know more, there are a lot in the books. Okay. So all these are all the inborn errors like histidinemia and all. Okay. But I don't expect you to know all. Just enough for you to know all the things that I mentioned: PKU, MSUD, homocystinuria, alkaptonuria. Okay, so those are the main important inborn errors of metabolism. Okay, any question on all this metabolism of amino acid? It's quite a lot. A lot of concept for you to understand, starting from the carbohydrate, lipid, and then amino acid. So tomorrow we are going to try to integrate all the metabolism. You can expect something like this, okay? And we are going to do it together. I'm not going to give lecture. I've already given you the slide for tomorrow, integration metabolism. So we're going to integrate all the carbohydrate, lipid, amino acid together. So I need input from you. Where is it? Yeah. So basically like this one, but this one also is quite simplified. So we're going to discuss more of this metabolism summary tomorrow. And I've also given you the practical. So during the practical, we will only discuss on the practical because you cannot do the practical too bad. So I've already given link on the how to use the glucometer and the urine dipstick. Okay. Hope that in future you have the opportunity to use the glucometer. Urine dipstick, urinalysis, you will learn later during the urinary system. So during that time also you can do the practical urinalysis. Okay. So during the class next week, Monday, so it won't be for 4 hours. It will only be like 2 hours, not even 2 hours, probably 1 hour. You can finish the practical. Just I'm going to explain the concept, principle and all, just like that. Okay, any question before I end the class? Okay, if there's no question, I see you tomorrow. We'll discuss on the integration of metabolism. Okay, thank you all.
Thank you, doctor.
Thank you, doctor.
Thank you,
Thank you,
doctor.
doctor.
Thank you, Doctor.
Doctor, how about the attendance?
Thank you, doctor.
Thank you, Doctor.
Thank you, doctor.
Attendance?
I think attendance I've already mentioned just now. The question is, what's the question just now? Name the enzyme that is important in homocystinuria, is it? Yeah. Name the enzyme that is important in homocystinuria. Okay. Thank you all.